name is Gabor Kovacs. I am a trained neurologist and neuropathologist. Neurologist means that I see patients at the clinic, and neuropathologist means that I evaluate tissue sections using the microscope, either from living patients in the form of biopsies or post-mortem if the brains are donated for research. I started my career in Hungary nearly 30 years ago at the neurology department, where I saw all kinds of neurological diseases. Very soon I recognized, and it was a bit frustrating, that especially for patients who had cognitive decline or movement disorders, we could not really offer therapy that changed their life dramatically or helped them. I realized that there is a huge diversity and every patient is different. And this led me to evaluate the neuropathological aspects, because we had a very famous neuropathology lab. So I started to go into the laboratory as frequently as I could beside the clinical practice. And this opened me completely new avenues, and I started to see why the patients are different from each other. And I also started to see the molecules that go wrong in the brains, which motivated me to carry on with these two types of careers, one a clinical and one a research. My main topics are, first of all, to develop biomarkers for living patients. In my opinion, to develop markers that diagnose diseases early, you need to use human post-mortem material, because that is where we see what goes wrong. So that is where we really can identify the targets for these biomarkers that can be used in living patients. So that is one of my focus. And second, I would like to understand why is this dramatic variability between patients. So I try to use and develop an unbiased type of regrouping of the diseases that can be then translated to the clinic. With this approach, I also think that we will understand better the therapy targets, that they must be very different for the patients. So altogether, my aim is to develop new stratification of diseases, new biomarkers that can identify these groups in living patients, and then identify the different targets that will be needed as therapy for the different groups of patients. One aspect of our studies is really that we had a breakthrough developing these tests for living patients, because we were able to reproduce the protein tests that we see in post-mortem brain. We were able to evaluate these in living patients in a tiny skin sample. So this was a very long, many years development, and we succeeded only recently, and we are leading in the world for the development of these type of markers for living patients. This is a very exciting. The other thing that we started to do is to implement so-called machine learning in our post-mortem evaluations, which means that we teach the machine to evaluate the microscopical alterations, and then we ask the machine to evaluate whether there is anything that the human eyes don't really see or notice. With these methods, we were able to identify new groups of diseases, which is very exciting for me. And this will be translated to the clinical practice, because again, it will help us understand why some patients have a very rapid, very fast progression of disease. Some patients have a very slow progression of disease. So with all these methods, we try to understand the new groups of diseases. And finally, we recently developed and use very novel type of microscopes. Just imagine that we are able to go down to really single molecules. Nobody has seen them before. And now we are able to see these bad proteins in a molecular level, what goes wrong in which cell type. And we will inform the clinicians and therapy developers to understand what really goes wrong in a human brain. And it will make and facilitate the therapy development. The other challenge that I see is the issue with collaboration. Although many people, many researchers emphasize the collaboration, I see that still as a challenge that the universities and different countries don't really come together to solve the diseases, but they try to do their own research and not always communicate or share. I see very positive trends that there is a pressure from the community to start a very strong data sharing, and I'm convinced that the data sharing, when it will open up a more transparent science, will dramatically facilitate the new developments. And finally, the next challenge that I think is that everybody is different. So this type of personalization in the therapy and the diagnosis we still have to bring into the practice. Currently, we are thinking of one disease or another disease, but every individual is very different. We call this individualized therapy or individualized testing for diseases, and that is exactly where post-mortem examinations can help, because we really show the spectrum of the variability of the different disorders. We just have to translate it for the clinical practice.